Alright everybody, welcome back to Lab Pathology 303, Lab 9. We only have 3 more labs, including this one, before we finish up the course. So, kind of sad, kind of excited about that. Alright, so today we're going to be working on fungal identification and alternate assignment for you remote students. So for you guys that are online, you may want to pay attention because I'm sure you guys have noticed that the slides that I have here during my lectures are a little bit different than what has been uploaded online. That's just because I edit my lectures to make it simpler for you guys to understand. However, for today, the information that we have though is completely different for you online students than what is posted online. So I'll see if we can get that replaced with what I have here today. So alright, let's get started. Just to quickly tell you what you online students have to do instead of this project that we're doing in class. You guys are going to have 2 assessments, 2 quiz type assignments. One part will be due this Friday and the second part will be due next Friday. It's going to be 20 questions each. Each of them is going to be worth 3 points per question. All the questions will be asked one at a time and must be completed before moving on. So make sure you answer the question and then hit next on the assignment because you won't have a chance to go back. So make sure everything's correct before you hit next. As I mentioned, you have until this Friday, October 23rd, to complete the first part of the assignment. You can use your lab manual and the weekly notes that are on eCampus, but you cannot use the internet or get help from your peers. So make sure you study up on everything before you take the assessment. Please don't cheat. Okay, so for the rest of the lecture then, it'll pertain to what we're going to be doing here. If you guys have seen these slides online already, these slides were produced right around spring of last semester when the pandemic first occurred and everyone had to switch to online delivery. So the slides that are online are narrated by Blake. She was also a graduate student, just graduated last semester, plant pathology as well. Most of the information that she says is what I'll be narrating today. It's just the techniques portion, except last semester it was relayed through a PowerPoint. So you guys can, again, just ignore the first part of the slides for you online folks about how the assignment is because it was changed. But for everything else you can go ahead and listen to it. It's just what I'm going to be explaining today. So last week, after we finished up all the inoculations, we had a little bit of time to transfer fungi or plugs of fungal cultures onto new media. The reason we did that is so we could start growing new mycelia so that we could examine them underneath the microscope today. As you recall, your project for you folks that are here in person is to identify the fungi that you have growing on your plates, and we're going to do that by looking at the mycelia, and specifically the hyphae, and as well as the spores. So I'm going to show you guys today how to make slides to observe the structures of the fungi and how to identify certain features that will help you key out the fungi or to identify them. So we're going to be making tape mounts and agar mounts. For those of you that are online, and also you guys if you want to see it online, Blake goes in detail on how to make slides for both agar mounts and the tape mounts. Agar mounts and the tape mounts, which I'm going to show you guys here, so I'm not going to play Blake's audio. Again, this is just more info on how to do it, and here Blake shows how she did it. More info on that. Making the tapes. Here's the important part. If you guys remember, a long time ago actually, we talked about the different types of fungi and how to identify them. We have aseptate and septate hyphae. Do you guys remember the other name for aseptate hyphae? Cenocytic, right. And that's one of the ways that you can characterize between the lower and the higher fungi. So your lower fungi are going to be your zygomycetes and your oomycetes, but remember your oomycetes are not technically considered fungi. However, they share many similarities with the lower fungi. And then you've got your higher fungi, which are your ascomycetes and your basidiomycetes. And looking at the hyphae, you can see here that some of the different fungi can have pigments, so they can be colored, and that's something you can use to help identify which fungus you're working with. You can look and see the septa and the hyphae, whereas over here with your lower fungi, it just looks like one large blob. And then probably the most important thing that you guys can look at are the spores themselves. This is why we transferred the fungi, the plugs of the mycelium to new plates so we can get more growth. With your lower fungi, the spores tend to be just single-celled, or at least just singular, round, not very complex looking. Whereas with your higher fungi, you have more more septations and more intricate structures of the spores themselves. You can also take a look at the colors, or the the pigment, I should say. So that's how you guys are going to be looking at your fungi when we make the slides and look at them underneath the microscopes. You want to immediately figure out whether you're looking at a lower fungi or higher fungi, and that'll help you to eliminate half of the possibilities just right off the bat. And that's really all we're doing today, so it's a pretty easy day in terms of, or pretty simple I should say, but it can be a little tricky. So any questions then? Hopefully you guys finish your project today for those of you that are online. Excuse me, for those of you that are here in person. You have until the end of next lab, so if possible, try to finish everything up today.
How are you wanting us to submit this?
The question is, how do you guys want to submit, or how do you want me to accept your assignments? So, for those of you that are here in person, what I'm going to be doing is just looking to see that I'm going to go around, because I have the answer key here to what all of your fungi are. You guys are going to draw and write down all your observations, and then you will grab one of these notebooks up here with all the different types of fungi on it and figure out which is which, and then I'll come around and sign your paper if you selected the correct one. And then after that you can either scan it or take a picture and upload it online with your assignment. Any other questions?
Next week we're going to have another lab to do, and then at the end we can finish working on our project.
Yeah, so next week we're going to have another lab that's going to be TMV observations, I believe. And if you have not finished by today, then you will have some time at the end of that lab. But after that, that's the cutoff. So try to finish up today is my recommendation. You guys have, what, 2 hours? That's what, 20 minutes per fungus? You guys can do it easily. All right? Any questions? Cool.